Okay, so say hello to the people online. There you go. All right, so this is, what day is today? Three? Period 5. This is Unit 10, Biochemistry. Okay, when I write this down, this is my second time writing this today. You suck it up and you write it all down. If it's up here written down, you write it down on your paper. I'm taking points off next time. And when I grade your test this next time, if I'm cranky when I grade it, I'll just take all the points. You won't get any credit for it.
What did you write under
Date,
unit?
period, unit, biochem. Okay, so we have B9A that we're still working on. And B9A says that you're going to compare the functions of different types of biomolecules. If you need to turn your desk around, do that. Lucas, if you need to move up, do that now too. You better get all these on there.
Wait, I got a question. Why do you rewrite it instead of writing it once and just showing it like?
Because a lot of people prefer that I write it with them. It makes more sense to them. I tend to go really fast if I'm not writing it down too, so that does that. So you're going to compare the functions of different types of biomolecules, including ... We're doing proteins. We already did carbohydrates and lipids, and we're going to do nucleic acids tomorrow. So I'm not going to write those down right now. So we're doing proteins today. And again, just like the other ones, we're going to talk about the little square here will be the third. It's the third of the biomolecule. So every time you see a square, that's going to be a biomolecule. There are only how many of them? 4. 4 that we're studying. There are more biomolecules than that, but these are the big dogs. These are the big 4. So for each of these, we're going to talk about the structure. There's your first circle, the function, and I'm going to give you examples. So the first thing here is the structure. And it's made of carbon, hydrogen, oxygen, and nitrogen. This is what makes it quite a bit different here. It's got the nitrogen on there. It's made of, what are proteins made of?
Amino acids.
There you go. So it's made of a chain of amino acids.
Does that say? Oh yeah, that does.
Okay, here's one of the big dogs right here, amino acids. And I'm going to get tired of writing it out, so I'm going to abbreviate it as capital A, capital A. And if it's plural, it's a little s beside it. So amino acids. So it's made of a chain of amino acids, and the order of the amino acids determines the function and the identity of the protein. So how do I abbreviate identity? ID determines the ID. So when you show your ID, you're showing your identity. You're saying this is who I am. So it determines the identity of the protein and its function. So the order of the amino acids is incredibly important here. So just like we've got 26 letters, how many words can we make out of those? Yeah, we have 20 different amino acids, 20 different amino acids that most living organisms use. And so out of all those 20 different amino acids, we get every protein possible that's in your body. Muscle tissue, color for your skin, whatever the colors are, different skin colors, things like that. We have eye colors that are different. All of those are different proteins. We have all the different hormones. You know, think about 60 to 75 different hormones in the body. So we have lots and lots and lots of different types of proteins. So we can get different words out of different letters. We can get different proteins out of different combinations of the amino acids. So that order of the amino acids tells us which protein it is, and it tells us how it's going to function. Because when you have amino acids building up, they're not just going to have 4 letters or 4 amino acids. They're going to have 1,000 of them. you know. So when you get that molecule and you've got all these amino acids in line, it starts wrinkling up on its own. It's called folding up on its own. And because of the order of the amino acids, it folds up in a different, are you listening? It folds up in a certain shape. Proteins, their function is in many cases controlled by the shape that they make. So you have globular proteins that are kind of blobby and round, yes? So they have different shapes and they have different functions because of those shapes. Yes? If you take organic chemistry when you get into college, if that's one of the roads you're going down, then you'll study this a lot more. But you just get a taste of it here and say, look, these are big molecules, and they're really complex. And it's not just a matter of what the name of it is or what the elements are that are in there. It's how it folds up that makes the shape. If it's folded wrong, that indicates to you that there's some kind of mutation that happened. It's not folded right. Usually if it's folded wrong, it's not going to work. So it has to be folded in a particular shape for it to work. The order of the amino acids is determined or made by the DNA. Yeah. We say that the DNA expresses itself as a protein. So your DNA is safe inside the nucleus, right? We don't want our DNA messed up, so it stays safe inside the nucleus. It's got the instructions. The instructions are to make a protein. So it expresses those instructions in the actual form of making the protein itself. All right, so we have the structure. Let's look at the function. Can I shrink this down a little bit? Can you guys still see okay?
Yeah. Running.
Oh, yes, move your desks down, please. Hey, don't talk to each other. Really? Okay, so function. So the function of the proteins basically make up most of your body parts. Muscle, hair, nails, anything that has color on your body, hormones, all your guts, your organs, stuff like that. All those things are made of protein. And they perform most of the actions in an organism. So when I say they perform most of the jobs in the organism, hormones, number one, you should think of. It's not just making a part like muscle tissue, but when we say hormones, remember that hormones is the second time you've written this down in this unit. Chemical messengers. These tell your body when to do certain jobs, when to do certain things, like take in sugar out of the bloodstream, stop taking in sugar out of the bloodstream, increase your heart rate, decrease your heart rate, take more water out of the blood so you urinate more, all controlled by hormones. Your cell membrane is made of a double layer of what? Okay, made of a double layer of lipids with big chunks of what? Proteins. So it controls what goes in and out of your cells. That's a function that it has. Antibodies. Anybody know what antibodies are? In general, something to do with what? Your immune system. Antibodies are chemicals produced by white blood. They're proteins produced by white blood cells, and they basically, you can think of them as like tagging something that's foreign to the body. Like if a virus gets in, Viruses have different proteins on the outside of them, and it's not you. Every single one of your cells has certain proteins on the outside of them, and your body recognizes that as you. This is me, this is not me. If we take blood from someone else and we transfuse it into you, first of all, if it's not the same exact type, you know that's a problem, right? It's got to be the same blood type. But even then, you can't take a lot of someone else's blood. There's a limit to how much you can take because your body understands that even though, yeah, it's red blood cells, they're not my red blood cells. And so you can transfuse some blood into another human, but there's a limit because the human's body you're putting it into will start attacking it because it's not their blood.
So is that why they have to find like people with the same blood type as me?
Blood type, first of all, is number one because the blood type is determined by the proteins that are on the outside of the blood. And these antibodies, if you put a new protein that's not, like if I put a protein that's not in my body into the bloodstream, it'll attack it. So it'll cause, if let's say I have AB positive. Okay, wait, that's a bad idea because I'm a universal acceptor. I can take all your blood. Let's say if you're O negative. O negative, you guys know your blood type? Okay, so if you're O negative, and we take his blood and try and put it into you, because his blood is positive, that means it's got this extra protein on it, and we put it in you, and your blood is going to attack that blood that's in there, and it'll clump up inside of you and cause the blood not to be able to flow through you. So, and it's because of that other protein. Antibodies will attack that. Antibodies are just chemicals, but they're spit out, they're created, and they cause them to all clump together and do that. So that's a bad thing. So if we have O positive and we take that and give it to someone who's O positive, then we have something where they can take the blood up to a point. There are other proteins on the blood cells, on your blood cells, that are not on someone else's. And so it'll take some of it, but after a time it's going to say no, it's just going to start killing that blood that you put into there. So a lot of times they'll transfuse blood, but then they'll also transfuse plasma. Plasma is the liquid portion of the blood to kind of help it keep the volume up, because you have to have a certain amount of blood in your body, right? Yeah. So they'll do a lot of that. They'll kind of put it together.
Draw your blood and see if you got COVID?
Yeah, that's probably one of the best ways to test, because they're looking for antibodies. There'll be chemicals in your blood that will say, hey, this chemical was specifically in response to the proteins that were on that COVID virus. It's a more effective test than the nasal test or anything like that. The blood test is the best test. So antibodies are proteins that fight disease. I'll give you a simple definition. There's more to it than that. Again, take anatomy. Well, then be ignorant the rest of your life. Come on, people. Hemoglobin. That's a really big, important one. Notice the word globin. That globin on it's a globular protein, so it's going to be kind of a rounded protein. It's in red blood cells. What color do you think hemoglobin is? Red. So it gives blood the red color and it carries oxygen. That's its job. It carries oxygen. That's its job. It just happens to be that when it carries oxygen, it's really bright red. Hemo, H-E-M-O, globin, glob, ion. There are these proteins, I'm going to go ahead and write the name. These proteins called histones. And they bind the DNA into chromosome form. Don't quote me on that word histones. I need to go check that. I think they're histones. Bind the DNA into chromosome form. You remember that the DNA is typically in chromatin form, where it's loose and spread out like a spaghetti, and you can't see it through the microscope when it's like that. And then during mitosis, it's got to be bound down into little chromosome form, right? Well, the thing that holds it all and packages it all together are little proteins, and I think they're called histones, but for some reason I'm doubting myself. They bind the DNA into that chromosome form. Kind of like if you ever have people moving and they load up the back of the pickup, the bed of the truck with stuff, and then they put those bungee cords on them to hold it down, or ropes on them. That's kind of what these proteins do. They're bungee cording the chromatin into the chromosome form to hold it all together. The next one would be enzymes. These are hugely important. Enzymes. Enzymes are biological catalysts. Okay, here's a word you're going to have to know. Catalyst. Catalysts are chemicals that speed up chemical reactions. without being used up. So catalysts are really cool because if we didn't have catalysts in our body, it'd be like you were in slow motion. Everything would be in slow motion. It might take you a year to walk across the floor. Okay, uber slow motion. Because we have catalysts, things happen more quickly. In many cases, things actually kind of slow down. Like when we do C6H12O6 plus O2, that would be like you setting this piece of paper on fire. Obviously, when you eat sugar, it doesn't get set on fire in your body. We control the reaction. That's one of the reasons why it doesn't just happen in one step. It happens in a series of steps so that we can kind of control the energy that goes out. But enzymes, because we have to break it down into multiple steps to burn that sugar, right? Burn the sugar, that's basically what we're doing. Because we have to break it into steps, we need catalysts to speed those reactions up so that they can actually happen in a time that makes it reasonable for us.
So if you were the only one with catalyst and like everyone else didn't have any, they would just all be moving slow.
Yes.
Super slow.
Yes, probably dead, but is that what?
Lost.
I don't know about their body. I know they don't have much in the way of muscle tissue at all, so they wouldn't require as much energy, things like that. So I don't know if it's because they don't have much muscle, then they don't require as much energy and they don't move as fast, or if there's some other biological thing. I've never studied them before. It'd be interesting. I know that a lot of animals like ferrets and birds and things, they have a lot more higher metabolism, so they're doing a lot more with catalysts, but yeah. It would make sense though, wouldn't it?
So would that mean, what would happen if you have too much catalyst? Would you be faster or would it?
Okay, so the catalyst is going to speed up the reaction, but there's a limit to what it can do. So it doesn't just, the more you have, the faster it goes. It doesn't work like that. Either you have the catalyst or you don't. So if you have the catalyst, it has X amount of rate of speeding it up. That's what we're studying in AP chemistry right now, like how do we calculate how fast the reaction is going, and with a catalyst or without a catalyst, and how do we do that? So at a certain temperature, it's only going to speed it up X amount. That's it. You would have to increase the temperature much more to make it go faster, even than that. So there are a lot of factors that play into it.
So it's kind of like an analytic converter. She doesn't. Don't you think it'd be similar? Wait, what is it? A cat. Oh, yeah. Yeah. Like,
So it speeds up what reaction?
the chemical reaction to add oxygen. It's on the exhaust.
Okay, so the deal is to be a okay, so a lot of times what they do when they make and name different parts, they don't necessarily go with the science that it's with. So in order for a catalytic converter to actually be a catalyst, you would have to say which reaction is it speeding up.
Well, it's not necessarily speeding up a reaction, but there is a chemical reaction.
Yes.
It's stuff on the inside of it that mixes with the exhaust, so it's less carbon dioxide.
So that it makes the emissions less toxic, right?
Yeah.
Yeah. So it's not, I think the word converter is more important than the word catalytic on that one because it alters the chemical reaction. It's like an excess chemical reaction, one step more, to make the exhaust not so toxic. So it's not a catalyst per se. Sometimes too, what you can go back when you see words like that, terms like that, you might go back to the original person who created it, and maybe they did have a catalytic reaction, and that's where they came up with the name for it. But sometimes they come up with names, and you're thinking, why did they use that name? Yeah.
Wait, so if you're born without catalyst, how like slow are you?
There are so many reactions, you would have to look at each one, but the problem is that there's a limit. If you're not born with those, you're just not going to be born. You're not going to make it.
Yeah.
Because it even starts right at the beginning. Because when you're just, a fertilized egg, and then you have 2 cells, and then 4 cells, and 8 cells, all those things are happening inside you. Even those cells are carrying out cellular respiration and all that stuff. So you have to have that process going on. You have to have those catalysts to survive.
I don't know, but like, let's say you didn't. How slow would you be?
So you're just asking how much it speeds it up. You would have to look at all the reactions that are going. You remember that big poster I put on the floor? You would have to look at all those reactions, and each of those reactions have a speed. And then what you would do is you would look for what's the slowest reaction, the one that limits it, and that's the one you would have to look at. I have no idea what the slowest reaction would be without a catalyst. Yeah. All right. The next one we have, would be the fact that another function of the protein is that it stores materials, which is weird to think of. And let me explain this really briefly. It stores chemicals that are needed by the organism, and usually these chemicals are toxic. So iron, pure iron, is toxic to you. So when your body gets it, has to have it. When your body gets it, it stores it in another compound, like within a compound. It stores it as a part of another compound. And a lot of times it's protein that stores that excess iron in. So you have it there when you need it, but it's not just straight iron because that's going to be poisonous to you. So it stores materials needed by the organism.
Aren't there different types of poison?
That may be toxic. There are lots of different types. We're going to use some tomorrow.
What's the most deadliest poison?
I think if you talk about coming from animals,
No, I'm talking about just all the poisons. Which poison like has
Yeah,
the
I
fastest
think-
effect?
oh man. Okay, so it depends on how much you put in. Jeez, there's so many. So it's needed by the organism that may be toxic, so that's why it's storing it. So, I mean, if you talk about just the old standard cyanide, if you put enough in someone, it's pretty fast, within about 30 seconds to a couple of minutes.
Let's say, what's the average amount? What's an average amount?
Of cyanide?
Toxic into somebody's body.
It depends again on the chemical, because some of them it may be 2 grains of it is enough to kill you, but it won't kill you immediately. Some of it may be 5 grams. You know,
Okay,
just,
let's say
just depends.
you just put 10 grams in.
Yeah.
Out of all the poisons, you put the same
Yeah.
amount. Which one would work best?
Yeah, I got nothing. You find that and tell me tomorrow.
Okay, Jacoby, can you help me? Okay, what about some animals?
I think there is. You guys might know this. I think there's a jellyfish, a little bitty tiny jellyfish, that's considered one of the most toxic. You guys, the most toxic animal, isn't it? One of those little. I don't know if it's a box jellyfish or something. Yeah.
No, those are huge.
Yeah. Okay, focus. So back on here, the proteins. Here's the key. All right. So put a little 2 stars beside this, and the reason I'm going to put 2 stars beside this is that there's a phrase here again. Proteins are the expression of genes.
How much electricity can we?
Okay, hang on. So you need to write your questions down in a list, and we'll talk about them later. All right. So proteins are the expression of genes. Focus. Protein. Did you write this down? You didn't. Write it down. That one. Okay. So when you talk about this, when you have this phrase, what you're talking about here is that the DNA, the genes. So when I say genes, you need to automatically think DNA, right? They have to stay safe inside the nucleus. So they have all the recipe and everything, all the ingredients are in there, and then they send it out into the cytoplasm, and they create, the instructions that they send out create the protein. So it expresses, the protein is the expression, it expresses what the DNA says. Yes? So have you guys ever been to Olive Garden? Yes. Okay. They have this dessert called, is it chocolate lasagna? Holy moly, it's good. So if you have all the ingredients for that chocolate lasagna, and you have all the recipe, so here's the recipe, here's the ingredients, the expression of that is the actual dessert that you made, the chocolate lasagna. Yeah? So the DNA is the instruction, and the pieces within the cytoplasm and the nucleus are the ingredients, but the final expression would be the protein that is made. So the proteins are the expression of the genes or the DNA here. The last thing on here, let's do some examples. Write this down. That's you I'm talking to. Write it down. Examples.
I couldn't see it. That's all I have to write.
Keep up with me. Okay, so, and I'm going to give you really some examples of enzymes because these are really some of the most important ones that we talk about, enzymes. So I'm going to put DNA helicase. It's a weird one. And we're going to talk about these a little bit later on when we talk about protein synthesis and when we talk about DNA replication. All right, do you know if you have OSE on the end of a chemical, it means it's anybody remember? A sugar, right? Like glucose, maltose, fructose, all those. If you have ASE on the end of a word, it means that it's an enzyme. So DNA helicase breaks open the DNA strand. What's the shape of a DNA? Double helix, helicase, yeah, you get that? So it breaks open the DNA strand, whether it's for replication or for protein synthesis, it breaks open the DNA strand. DNA polymerase, polymer, ACE, actually breaks it open so that it can add a nucleotide. Okay, have you guys heard the word nucleotide before? Nucleotide? Okay, so DNA, okay, so monomer and polymer. Monomer means there's one molecule. Polymer means you take that one molecule and you duplicate it, or you add individual pieces to it. So polymer is made of multiple monomers. Monomer is one, polymer means you've got chunks of pieces of that. So DNA is made, it's a polymer, it's a huge polymer at all. And this DNA is made of nucleotides. So proteins are made of what?
Amino acids.
Liberty, are you with me here? Proteins are made of what?
Amino acids.
Amino acids, right? And each amino acid is just a monomer, but when you put them all together, you get a polymer. The things that make up DNA are called nucleotides. So that's what it's talking about. So this DNA polymerase is a protein. It breaks open that DNA and enables it to add another piece to it. And that happens when you're replicating, when you're making copies of the DNA. Another one would be amylase, which is in your spit. What's it good for? Breaking down starches. And then one more, and it's going to be more of a generic one, detergents. Okay, so in this day and age, almost every detergent you pick up has enzymes in it. Enzymes, proteins, and what they do is they can break apart stains. Like if you spill ketchup all over your shirt, it breaks down the ketchup. Now, the problem with enzymes is that they are easily messed up. They are easily broken down. And so if you wash all your clothes in hot water, you have totally wasted your money on detergent. Any of you guys wash everything in hot water? That's a good thing. If you read the instructions on the detergent bottle, for those of you who have never done laundry, you have instructions on the bottle. If you read the instructions, it says warm water or cool water. Because when these detergents, they have enzymes, if you put them in hot water, they don't work. They can break down organic stains, grass stains, food stains, mustard, ketchup, all that good stuff that you guys drip all over yourself. So it contains enzymes that break apart stains. So, but if you put them in hot water, it denatures the protein, it messes the protein up. Anytime you put proteins in hot water or treat them to high temperatures and stuff, it'll mess them up. Once you've written that down, I want you to flip over to page, I think it's 47 in your packet.